Hello, Professor and classmates. My name is VU THI BICH NGOC. I come from Plant Biotechnology Lab. Today, I would like to present my research report named Upstream Open Reading Frame (UORF) containing BZIP68 mediated integrated stress response (ISR) in plants. As you know, the cells can be exposed to various environmental stresses such as high temperature, drought, salinity, viral infection, light, or herbicide. These kinds of stimuli can lead to accumulation of unfolded proteins in endoplasmic reticulum (ER), resulting in ER stress, then induced adaptive mechanism known as UPR, unfolded protein response. In plants, the mechanisms of UPR signaling pathway are less well studied. In recent years, two signaling pathways already has been reported. First one is IL-1. Second pathway is BZIP16 and BZIP78. You can see in the figure, unfolded proteins will binding to BIF binding protein and activate two pathways, resulting in induced UPR mechanism to restore cellular homeostasis. However, one pathway also looks like existing in plants. This pathway is known to integrated stress response. In response to cellular stresses such as ER stress, amino acid starvation, viral infection, iron deprivation, to restore cellular homeostasis, the cells, including plants, can trigger ISR, integrated stress response. This pathway is the phosphorylation of eIF2 alpha by one of four members of eIF2 alpha kinase, IL-1, GCN2, PKR, and HRI. Integrated stress response can effect inhibition of almost general protein translation through eIF2 alpha under ER stress. However, there are some selected genes, including ATF4, is well known as responsive gene. ATF4 is a transcription factor in mammalian cells, thus containing UORF, upstream open reading frame, in 5' UTR, can increase protein expression by eIF2 alpha phosphorylation. Plants don't have PRK, PKR, and HRI, but plants containing GCN2 and eIF2 alpha regulated UPR. This pathway looks like conserved ISR in plants. That's why we are looking for a gene as a candidate of ATF4 existing in plants and have a kind of characteristic. This can be regulated by their protein. In my research, we perform in silico analysis to investigate functional autologs of the ATF4 and found that some transcription factors like Arabidopsis, BZIP16, and BZIP68 belong to Z group that containing upstream ORF in 5' UTR are potential candidates. Previous study indicated that BZIP16 is negative regulator in light-mediated inhibition of cell elongation, and BZIP16 is positive regulator in light-regulated seed germination. In other papers, they reported that BZIP16 and BZIP68 are closely homologous and they are known to interact with each other by just two hybrid assays. However, The biological function of BZIP68 is not well understood. We just know that BZIP68 belongs to Z group, BZIP family. BZIP68 also closely homologous with BZIP16 and known to interact with another gene like BZIP16 and ZBF1. In our study, we use qRT-PCR to investigate whether BZIP68 and BZIP16 is related to ER stress response. We examine the expression level of BZIP16 and BZIP68, including VU3, as a representative marker genes for ER response under prolonged ER stress using molecular analysis qRT-PCR. In this experiment, plants Co0 were treated with tunicamycin, the chemical induced ER stress condition, for 7 days. The results show that VU3 is highly induced after 3 days and increased again at 7 days by approximately 2,000-fold, same to previous report. Unlike VU3, the expression levels of BZIP16 and BZIP68 were slightly different. They reached the highest 2.5-fold at 6 days, then subsequently decreased after 7 days. The results suggesting that BZIP68 transcription slightly increased during prolonged ER stress. However, we don't know it's enough for ER stress response or not. Then we characterize the biological function of BZIP68. We obtain 2 DNA insertion BZIP68 mutant and confirming by PCR. And we make overexpressing BZIP68 using 38 promoter and fusion with the FP tag. In the figure B, in normal conditions compared to wild type, the overexpression line BZIP68 didn't show any difference, whether in tunicamycin induced ER stress conditions. Most of overexpressing plants show different phenotype compared to Co0. However, the sensitive phenotype is different according to the light. We select 2 lights, 20+2 and 22+3, show more strongly sensitive phenotype as 2 representative lights and rename them as BZIP68 overexpression 1 and BZIP68 overexpression 2, then confirmed by Western blot to see the FP signal, and really, 2 lights show more strongly the FP signal. For this experiment, we compare whole plant phenotype of BZIP68 overexpression line together with Co0 and knockout mutant in MS media and tunicamycin media. We can see that the results show overexpression lines are more sensitive in ER stress than Co0. It means that gene transcription factor BZIP68 related to ER stress response. These are some methods as qRT-PCR, Western blot, or phenotypic assay we use to determine function of the gene BZIP68. Thank you so much for your attention.